Okay, so welcome. I just got here last night, so this is a bold thing to be doing with jet lag. Nevertheless, it's a pleasure to talk to a captive audience. I do it all the time. I usually use props, so we'll see how this goes. What I will attempt to do is at least perhaps the first two-thirds. I can focus on the so-called learning objectives. So my goal is, if you are not familiar with the antimicrobial resistance crisis, what phage therapy is and why it might be useful in combating this, and how a particular approach to it could be especially useful, and this is a way to steer evolution down a preferred path. So I'm being purposely vague because otherwise I don't have any punchlines to give you for these. So we'll get there. But I guess the first thing to say is that the remaining third, if I get there, and it's okay if I don't get there, is that this is kind of familiar territory for maybe half the people in the room who are here for the program that I'm participating in, either because they've suffered through my talks before or they just are knowledgeable on these topics. If time, I'll talk about something more provocative that, not to say everyone in the room is a stranger to, but there's lots of things that, this constitutes a very applied problem that I work on, but there are plenty of other problems I work on that are not so much applied, they're just conceptual, and they involve aspects of phage evolution on planet Earth, sort of a grand scale, and we'll see if we get there. Antibiotics, beginning with the first bullet point. So many people, if you stop them on the street, they know that antibiotics are useful. I mean, what they really are is that it's a great example of how humans found something that has been present in the biosphere, and then we leveraged it to our advantage. So antibiotics, you could think of them as largely chemicals that are produced and secreted by bacteria, also by fungi, and the purposes that they have in terms of kind of interactions between microbes, what we care about the most is that these antibiotics can be lethal. So one antibiotic could be secreted by a microbe. Consider if it's under competition with another microbe. They use this to their advantage and they kill their competitor. So in 1928, I guess I'll put that up here as an important timestamp. In 1928, very famously, Alexander Fleming kind of discovered them by accident. You can grow bacteria and fungi and other, well, primarily those on agar plates at this time, and you could not be so clean as to clean up your own experiments. You can go away on vacation. I think literally this happened. And Fleming comes back and he starts to see these zones of inhibited growth on plates. And this was the aha moment to say that, at this time, you have to remember there was a lot of mortality in the human population due to bacterial infections. And so this was seen as a wonder opportunity to harness something that you can scavenge from certain microbes. They're growing, they're excreting these. We scavenge them, and we start using them in the clinic to kill bacteria purposefully and solve infection problems. I would say that plus the advent of cleaner water in many parts of the world, not all parts of the world, access to antibiotics and cleaner water caused infant mortality, childhood mortality to plummet. And so you had this amazing benefit of something that could be produced, you harvest it, and you deploy it. Okay, so not long after, the problem is Fleming discovers penicillin, and then others are looking at other compounds that are very successful, and they start to see a problem with this eventually. Not immediately, eventually. So in the places that really took this technology, used it in the clinics, the hospitals, etc., saved lives, as I've said, this has saved countless human lives through time. And the punchline is it doesn't work that well anymore. Okay? So kind of a snapshot of such data in places where the data are closely tracked. You can imagine, through time here. And the data set I'll show you just happens to be from Finland. in a study where they were looking at the frequency of, say, prescription antibiotics used between, I don't know, it was like 1982 roughly, and perhaps the mid-1990s, like 1995. And antibiotics do work, and if you focus in on some population group, such as kids with ear infections, and you're trying to target the bacteria that cause them, you can get access to all the data if you are scrutinizing it in one geographic location, and prescription antibiotics rise through time. Why? Because it's useful. So I'm not going to say what it started at, just in this time period, it rises. So the problem is, in the same patient group, you can look at the frequency of antimicrobial resistance, same time periods, 82 to say 95, and that occurs. So this is best explained by natural selection. So you've got bacteria in a particular environment, and that environment is ear infections. And the situation changes such that they are now exposed to something that is lethal. So this produces strong selection pressure for the populations to rely upon mutations that occur spontaneously, and those variants will take over the population. So you start to get this effect, that the antimicrobial resistance rises, and let's just say, for the illustrative purposes of this, imagine that suddenly this drug used on this type of bacterium in this patient population becomes useless. Okay? Now, not all geographic locations and patient groups have this kind of scrutiny of the data, but if you can imagine this happening globally, that's the problem. If you just looked at the big picture of antibiotics have been used prevalently, ever prevalently through time, in the clinic, in hospitals, because as I've said, we've turned to them to save lives. We also use them a lot, even more so in agriculture, animal agriculture especially, where you could imagine farmers using them prophylactically to protect their animals. Turns out that antibiotics given to animals that we harvest for meat make the animals plumper, and you get more meat per animal. So there's a lot of drive in farming practices to also use antibiotics at very, very high rates. So all this counts up to kind of a large, uncontrolled experiment being conducted around the world, where we're changing the average environments that many bacteria experience, and we're selecting for antimicrobial resistance to the extent that there are plenty of bacteria now that we worry that they are multi-drug resistant. So they're not only gaining resistance to, say, penicillin, they're also gaining resistance to things that act a lot like penicillin, or could even be drugs from different drug classes than penicillin. And in the end, it could really go off the rails, and you get something like pan-drug resistance, or some target bacterium, where every approved antibiotic no longer works anymore. So that's like the very sobering outcome. And when that occurs, then you have to really turn to other technology for solutions. But the main point of this first bullet point is that antimicrobial resistance is on the rise. If you go to the hospital for some trivial problem, increasingly surgeries are actually pretty easily done and quickly become outpatient. And then you end up being in this hotbed of possible infections for things that are kind of literally infecting the hospital, and you unfortunately catch this infection, it's multi-drug or it's pan-drug resistant, and then suddenly your situation as a patient radically changes. You go in for some surgery, during the surgery or after the fact, that you're recovering in your hospital bed, that environment places you under exposure for something that is a bigger problem than what you entered the clinic for. And the worst case scenario is if there are no solutions to help you. And it becomes a lethal occurrence of you having to go in for some surgery, but a byproduct effect has taken your life. So any questions on that? Is everybody okay with that? Huh, remarkable. All right, so if antibiotics don't work so well anymore, the question is, what are you going to do about it? So there are lots of technologies that are on the rise to potentially solve the problem. I'm going to go back in time now to an earlier time point, circa 1917, and the work of individuals like, especially Durrell, whose name I may not spell correctly, but this is one of the two people credited with discovering something that, for all we know, has been ever present on Earth alongside the evolution of bacteria very long ago, billions of years ago. There are genetic elements that exploit and attack and take advantage of bacteria. And the ones that I'm going to focus on now are called bacteriophages, or phages for short. Now that we're grappling with the pandemic that ideally we can completely solve soon, but apparently not soon enough, it used to be I had to explain to the audience what a virus is. Good news, bad news is that I don't have to explain it so much. The bad news is everybody thinks they're an armchair expert in virology. And there's a lot of misinformation out there. For the sake of this presentation, think of it as cellular life is very common on Earth. You have single-celled organisms like bacteria, you have multicellular organisms like us, but by and large, cellular life is exploited by viruses, which are acellular. They are nucleic acid, they are protected by protein and can exist outside of the cell, some of them. For the purpose of today, those are basically the ones I'm talking about. But certain of these viruses can only replicate on bacteria. And again, for today, I'm just going to refer to those as phages. And a specific kind of phage is what I'm interested in, where here's your bacterial cell. Imagine this is a phage, and it's trying to do its business to replicate in the biosphere by entering this cell and turning it into essentially a production factory to create copies of that phage, which then goes on, in this case, to lyse the cell. And these phages can exit and then come back here and infect similar cell types. And so these are called lytic phages. And this process of killing the cell is lysis. All right? So during the course of infection, replication of these particles occurs within the cell itself. You can think of literally the cell exploding and releasing these particles. And if there are other susceptible bacterial cells in the environment for them to infect, they can go on and continue that cycle. So note that in 1917, which predates Fleming, this was discovered. So you had to have good enough equipment, eventually, in microscopy, to even see virus particles, right? This is amazing that microbiology, in the backdrop, been studying it for a long period of time, up until the early 1900s, but not appreciating so much that arguably what dominates the biosphere, numerically, are viruses. Biodiversity-wise, maybe also, we can talk about that. The fact that I'm trying to put forward is that soon after Felix Durrell co-discovers phages, he starts immediately using them in experiments to see whether they can conquer infections in animal models and in human subjects. Because it was back in the day when you could do experiments on humans, essentially, which is ethically out of bounds now for very good reasons.
Sorry, in which country was he working?
In which country?
Yeah.
This work, a lot of his early work was done in, I think, Canada and the US. He had only one academic appointment in his career, and that was at Yale for about 5 years. He is very famous for, not long after this, working with George Eliava in the USSR to develop and launch an institute for phage therapy that still operates today, the Eliava Institute. Turns out that Stalin executed Eliava as an enemy of the state. I can't remember the exact reasons why. I'm sure the Eliava family cared, and they knew why. But the point is, unfortunately, then that soured the relationship for Durrell, I think, with the USSR, He ends up doing a lot of his work outside of there. The point is that this was very much in the backdrop because antibiotics were dominating the focus of Western countries. And in places like Russia and former Soviet republics, especially the country of Georgia, where the Eliava Institute still operates, they've been studying phage therapy since that time. In the USA, if you ask somebody what is phage therapy, maybe they start to hear about it for reasons that I'll get into in a moment. But largely, the US and other Western countries stayed away from doing a lot of phage therapy research because of the Cold War. It became a geopolitics issue somewhat to the thinking of, well, that's Soviet technology, why would we want to do that? Now, unbeknownst to you who are US citizens, there was plenty of phage therapy research going on in our military infrastructure. That's probably not that surprising, in order to keep pace with the Russians. So why I'm going into all this is that this old technology, which was seemingly kind of less elegant, you're using something that is a biological drug rather than something that is more easily controlled during production and trusted to be, that's penicillin and nothing else is that. We're getting desperate. So I would say that turning our attention back to something that was seen as rather amateurish is now happening in earnest. And there are plenty of startup companies, somewhat big pharma is starting to get on board with this a little bit, again out of desperation. But groups like mine are trying to take our phage biology expertise to this problem and update it for modern times, because of one big issue with phage therapy that I can now get into that is a problem similar to what I'm erasing. If you consider phages being present entities within the same environments of bacteria, for all we know, for billions of years, you would expect that there are plenty of defense mechanisms that a bacterial population might have against phages that are attempting to infect the cell. So some of it is a block at entry. So this very proximate problem of any virus that's existing outside of a cell has to recognize the cell in order to initiate an infection. This is a very proximate problem, I would say on average for any virus. That was a bit of a bold statement, but I'll stick to it. So imagine if this phage is able to gain entry into the cell, well, there might be other blocks within it, such as restriction modification systems, which are ways that enzymes will recognize certain stretches of foreign nucleic acid and cut it at that region and stop the infection from proceeding within the cell. More recently, elaborate defense mechanisms, oops, misspelled that, CRISPRs, which are essentially like adaptive immunity of a sort that bacterial populations can kind of recognize familiar territory for the phages that are trying to exploit them, and then create an array that allows the average cell in the population to recognize that phage if it enters, which is a lot like your adaptive immune system. So with these many defenses that are in place, or that evolution could cause bacteria to improve if phage are selective factors in their environment, there's a lot working against phage therapy to even proceed and be useful. Okay? So we have more and more knowledge of these literally every day. There are some research programs that are devoted to looking at new defense systems that have not been described yet, but occur in bacteria. But I'm telling you honestly, and this is kind of one of the mysteries of the work that we do on this, is that phage therapy has worked very well through the years. Now, I already told you not that many people relative to others who are focusing on antibiotics were studying the problem. But there are studies and studies and studies over over 100 years now to show how phage therapy can work despite the fact that these defense systems exist. Nevertheless, the expectation is if you're in the hospital and you have some bacterial infection that's multi-drug or even worse, pan-drug resistant, and I'm going to deploy phage therapy somehow to try to alleviate your problem, I'm going to impose strong selection pressure on the bacteria to overcome that. And these defenses, let's say they were not working initially, they can quickly evolve to work better. Provocative statement, maybe number one, that I'll stand by is that this is always going to be an issue in phage therapy in the clinic, is you should expect that the evolution of phage resistance, similar to the evolution of antibiotic resistance, is going to be a hindrance to moving forward.
What's known quantitatively about how good the resistance is, say, from CRISPRs, if they are exposed to a phage they've seen recently?
We're very interested in thinking about if you deploy a phage, and what I'm saying is true, you get evolution of phage resistance. Will it always occur similarly? No, because there's lots of mutations that could provide that solution. But some of your question, Daniel, is having me think about, I'll eventually get to, if the type of evolved resistance in the bacteria is largely of one set of mutations, and there are these other rare ones that don't work so well for the technology that I'll explain, maybe they're so rare I don't care. Okay, so I don't care maybe because they would occur so rarely that the bacterial population just doesn't find that stochastically from the universe of possibilities. Or those resistance mutations could have such a fitness cost for just growth that they become almost negligible to me. Okay? So I'm now going to, so keep that in mind. If evolution of phage resistance is always going to be at least potentially problematic, is there a way to develop the technology now that hadn't really been done in the past to alleviate this being a problem? So how's that going to happen? In the mid-1990s, I was working as a postdoc at the University of Maryland in Lynn Chow's group. And up to that point, I had been studying evolution of bacteria and their plasmids. But I wanted to start working on viruses. I took a very short leap into phages, because phages, as I've said, infect bacteria. And so in the laboratory, we saw something interesting. And I'm going to draw a cell, and these structures here that I'm drawing are pili. So imagine if this is a bacterium that is infecting plants. Plenty of bacteria infect plants. But the way that these bacteria can infect plants, I'll now attempt to draw a leaf. That went remarkably well, he says, as he's drawing it. So these pili, for many plant pathogenic bacteria, they allow movement. They're extended and retracted, and they allow something called twitching motility, where you can imagine the bacteria moving across the leaf surface. And if you recall from your Botany 101, there are holes in leaves called stomata. And it is possible for the bacteria to gain that as an entry point, to then infect the plant, flourish in the plant, and be a plant pathogenic bacterium, essentially using the plant as a resource. So these are absolutely crucial structures for these bacteria to use in natural environments to go about their normal way of life. So what we studied in this laboratory in Maryland were some phages that exploit these pili as the initial entry points to the cell. And they do this because the model is that they are attaching to the pili, which I'm telling you normally extend and retract. And when they retract, it brings these phage particles closer to the surface of the bacteria, and they gain entry. Without the gory details, that's all you need to know. In the laboratory, we weren't working on plants. Okay? We had no interest in working on plants. We were actually just working on phage bacteria interactions. And what can happen in this scenario is that natural selection can change the bacteria to this phenotype. So it favors mutants in the laboratory that just don't have pili. And they gain this strong resistance to phage attack. So this is the evolutionary resistance you can see in the laboratory. Makes good sense. Just eliminate the structure that would be important to the bacteria for their susceptibility to the phage. But of course, then this is the problem. They could not, if we challenged them to traverse a leaf surface and enter, they're not going to be able to do it. So it became immediately obvious that this is an example of something that's often seen as natural selection solves problems, but it doesn't make organisms absolutely fit in all environments. It's solving a proximate issue, but the trade-off here would be if these bacteria were in their normal environment, they couldn't get away with a set of mutations, which gets a little bit to Daniel's question, is that these are very easily obtained in the laboratory mutations. So the point is whether you could use knowledge of this to improve this problem of the evolution of phage resistance if I'm claiming it's always going to occur in the clinic. And that's actually this third bullet point. So far I told you antimicrobial resistance crisis exists. Might have impacted your lives, might have impacted your lives of your loved ones. I mean, this is a real issue. Phage therapy is old technology that in some ways could alleviate the effects that we're seeing in the clinic, and you use phages just as Durrell did back in the day, but we can't be so naive, because if you deploy the phages, you're going to just select for resistant forms of the bacteria to those phages. So we wanted to take this observation and leverage it, because what I drew here is that pathogenicity can be compromised when a bacterial pathogen is interacting with a phage. It cannot have its cake and eat it too, is basically the prediction. That there's a steering that the phage provides, that if the mutations that would cause the bacteria to give up pathogenicity are prevalent enough to solve the phage problem, you kind of paint the bacteria into a corner. Okay? All right, so think of it now. Let's try and think of it this way in the clinic. So, all right, clinic here. And this is a little bit more of a state space. But if there's some virulence or pathogenicity trait of a target bacterium, and that's increasing along this axis, think of it as at odds with, this is, well, I guess I should write it out, phage sensitivity. So let's say at time 0 in the clinic, you enter the hospital, you have a dangerous bacterial infection, it's highly virulent, it's causing you a lot of disease distress, maybe threatening your life. The goal is, if these bacteria are known to be highly sensitive to phages that we could use in the clinic, then ideally what's going to happen, well, here's the bad outcome, is that through the course of time, somebody is trying to control these with antibiotics alone, and you just get this occurring. So no phages are deployed, antibiotic resistance is on the rise in the clinic, that disease population exists within the body, goes forward in time, and maybe here is a mortality event, okay? Just try and draw a cross there. That's what's usually used in the medical field as mortality has happened. That's what we see increasingly in the clinic, and phage therapy ideally could perform a solution. So let's just say the wild-type bacteria pathogen has some traits that are associated with its virulence. I'm now going to introduce another pretty alarming trait that some bacteria have that we care a lot about, And these are protein complexes that span the inner and outer portion of the cell. And if antibiotics, we'll draw these as little circles here, if they enter the cell, they're actively pumped out. These are called efflux pumps, and they work very efficiently across different drug classes. So the first idea we put forward to examine if you have a virulence trait that's problematic in the clinic, and if you want to have phage therapy provide a solution that drives a trade-off, find some phage that is interacting with those efflux pumps. Because the bacteria have a very proximate problem. They're dying of phage attack. And something that they can do is strongly alter the structure or just get rid of the efflux pumps. Okay? So in this sort of state space, ideally, we're trying to drive them to this corner. Or imagine this is the clinical breakpoint. So you're basically taking a bacterium like this, and I'll draw it again, where it is phage resistant. So here's sensitivity, here's resistance, and I put it down into this corner, but you'll notice it has no efflux pumps. So what happens then? You deploy an antibiotic that had been useless, and it's suddenly useful again. Okay? That's an important punchline. Let me repeat it. It means that if you have bacteria that have some mechanism of antibiotic resistance, you could put them in a phage environment, essentially by deploying phages in therapy, and change the bacteria without necessarily eliminating them, by steering them down an evolutionary path of phage resistance that I prefer and that the patient will benefit from in the hospital bed. Okay?
If we're given antibiotics, do they only have one kind of efflux pump, or can they have many different kinds and the phage only binds?
It's been worrisome to observe that efflux pumps work well across drug classes. Maybe that's what you're asking.
No, I was about several efflux pumps for the same antibiotic.
Yes, I'm not as much of an expert on that, but there can be an intimacy of efflux pumps that perform lots of functions. We just care about them in the clinic because they're pushing antibiotics out of the cell. So I think there are probably experts in the room on that. That ain't me. But I happen to work on efflux pumps in certain target bacteria that are increasingly worrisome for causing these drug-resistant phenotypes in the clinic, especially Pseudomonas aeruginosa. Yes?
Are there general differences in specificity of antibiotics and phage? So, my intuition might be that antibiotics tend to work across a broader range of, you can have broad spectrum antibiotics, but I don't know such a thing as synthases.
Yeah, so let me say broad spectrum antibiotics versus narrow spectrum are both broader in their action in killing bacteria than phages tend to be. Phages tend to be pretty specific to the target species, if not the genotypes within the target species of bacteria. Some exist that kill across species. They don't seem to be that common, or we're not good about isolating them. That's a long-winded answer to your question. Essentially, antibiotics are broader in their killing, which is problematic for your microbiome and the gastrointestinal distress that you often have when you take antibiotics. They're killing the thing they're supposed to kill as a pathogen, but they might also be killing the flora that are your helpful commensals in your gut. Does that help?
Yeah.
Okay.
So phage therapies in some cases will then have to be tailored to the particular...
Okay. So now you're asking the money question. So the development of phage therapy has often been concerned with specificity is a good and bad thing. If I deploy a phage and it's highly specific to the target, I don't have to worry that much about off-target killing. The problem is if all of you have a Pseudomonas aeruginosa infection and it's infecting the same organ in your body, you might have a lot of genotypic differences among those bacteria. And the phages that I would want to deploy, it might be problematic for me to find a phage that kills across that genotype space. That's almost impossible, I would say. But, and then we could quickly get into what are called cocktails, where you can mix different phages together and get a broader killing across your genotype space of the bacteria that are causing you disease. Yeah? Correct. So would I be, so the doom and gloom scenario would be, and let me take your comment to the nth degree. If I used phage therapy very broadly across Earth, would I select for the same problem that the antimicrobial resistance crisis is already creating? Wrote an opinion paper on it because I was sitting around during the pandemic doing things, and I would say the answer is no. It's because of the way that bacteria share antibiotic resistance genes with one another, often through, it can happen on the chromosome, but they'll often have plasmids, which are miniature chromosomes that are not, they're accessory. They're not always essential for the bacteria, but they can easily move from cell to cell, even across different species. So that has contributed to the rise of antibiotic resistance. Resistance to phages doesn't tend to work that way. It just, I can get mechanistically into it, but it's not that they're frequently found as resistance genes on plasmids. It's basically chromosomal. And it's just not this expectation that the selection for phage resistance at a global level would at all be anything like we see with the antibiotic resistance crisis. Okay? Yeah.
So I want to ask about this sort of this trade-off idea.
Mm-hmm.
So this, if I'm not mistaken, this seems to presume that the solution space that the bacteria have at hand for dealing with phages is fairly low dimensional.
Yep.
So that if you give it all the right dimensions, it can get out. Is there a reason for this?
So, I guess to your question, imagine if this is a distribution of, I don't know, genotype space, and what I'm claiming / hoping is that the bacteria will solve the phage problem with a vast majority of strong trade-off driven solutions, and that there'll be this little portion of this spectrum of possibilities that would be contrary to my prediction. And the question is, do I care? I may not care if these solutions exist, but they are very difficult ones to spread in the bacterial population because the cost of them is too high, or the mutations that actually occur are very rare compared to other mutations that are more easily achieved in the bacterial population. But essentially what you're saying is true. I can't control this in the clinic. What I haven't told you so far is we've actually done this, and it's worked most of the time, including the recent data that we have that I go into a patient, I take samples from them longitudinally, and I look at which bacteria are left over after the phage therapy has been deployed. And there are these mutants that suffer trade-offs that I can measure in the laboratory. So that's good news. The bad news would be for 1 or 2 patients, or whatever it is, where the bacteria will solve the problem by not gaining this trade-off. So that would be the bad outcome, especially if it was a pan-drug resistant bacterium to all known antibiotics, and we're using phage therapy as a Hail Mary pass, and it's just not working.
But so if I take naive bacteria and I treat with antibiotics and I look for the first resistance or tolerance that comes out, these are all often also trade-offs.
True.
And,
Yeah.
And it's only later that they come up with the resistance.
Yeah, so you can think of that sort of ratcheting up and down in bacterial populations across the decades. But they've now settled in a state space where they're not suffering these problems. They've got plenty of drug-resistant genes and can gain plenty more through plasmid acquisition, and we're left with the problem in the clinic that I'm talking about. Wow, this is great. Okay. I'm not going to get beyond this, I have a feeling. Yeah, Ariane, yeah.
So how well characterized are the phages that are used in phage therapy?
So do you have single lineages of which you know that have been screened for effects on different bacteria?
Wait, are you asking which phages to use?
Yeah, how well characterized are they?
Ah.
I just have no clue.
Okay. A hell of a lot better characterized now than they were in the past. So imagine it's the early 1900s, and you're developing phage therapy, and what you are benefiting from is something that was determined much later on. You literally could go in the soil outside of your, I don't know, your house or the river in your neighborhood, and you could screen for phages on the target bacteria that you're trying to kill, and you'll easily find them depending on what those bacteria are. And then you start using them. That is an example of like no characterization, right? We can't get away with that now because the U.S. Food and Drug Administration wants a whole lot of check the boxes on, do you know this, do you know this, do you know this, okay, good. The patient is out of options, give them a waiver, they signed it, done. So what shocked me is that that's what we did. And we killed no mice before we started deploying phages in emergency therapy in patient cases in our hospital and other ones. Why? Because there's a desperation for it. And when you talk about the antibiotic resistance crisis, usually I can't draw it because it would be too detailed. You often see these sobering projection maps. Go out to the year 2050, and if the rate of the problem occurring continues to be increasingly bad news, then suddenly AMR becomes a leading cause of death in humans again. And you start to get back to the pre-antibiotic era, which would be alarming, and it would be evidence that you could have this recurring problem cause more deaths annually than cancer. And cancer is on the rise. So let me state strongly, I'm not believing necessarily in the projection maps. I am believing in the problem that physicians bring to my attention is that some people are already in crisis mode because they're immune compromised or they're just highly vulnerable to bacterial infections. Those are the people signing a waiver now who are willing to be experiments. Boris.
So going back to this question of specificity, so there is a notion, so you just explained, so if you want to find the phage killing particular bacteria, so you just pour some river water in the liquid. But how often in these clinical problem cases does one know the exact cause of infection? You know, with the last time I was taking antibiotics, nobody figured out all this bacteria was killed.
Right, because you weren't at death's door. So there were some suspicions. So if you couldn't hear it, the question is, how sure are we of the bacteria that cause an infection in the first place, such that I would be able to find a phage that is characterized and use it in emergency settings? So the clinical microbiology is improving through time, okay? But it is not perfect, nor is that money going to be expended necessarily if you have an annoying problem as opposed to a death's door problem, right? So I'd say there's a range of how that is examined for you in your case. But maybe the nature of your question is from the specificity of the phage attacking the bacteria, and I'm not sure what the bacteria are that are causing your illness. Ideally, phage therapy might graduate to being good enough predictive power about what your bacterial illness is. Here's the menu items that I have for phages. Oh, okay, he's got this. Oh, let's give him these 3 phages. And it will be the same as, let's give him penicillin and maybe amoxicillin and some other things that we think would kill your bacterial infection, and then hope it works. Okay? So I would say for drug labeling, and when phages eventually become labeled as drugs, in the USA, because they're already labeled as drugs elsewhere, I can get into that, then ideally we're getting closer and closer to the clinical microbiology matching what we think would be a library of phages that would be useful.
So if I understand your answer correctly, you're saying that the path forward that you see is in this broadening specificity of phages, approved or
Yes.
phage cocktail, as opposed to improvement in diagnosis. Because I hear
Yes.
about sequencing snippets of DNA from the blood and determining the bacteria that's harbored somewhere, right? Doing diagnosis, right? There is potential improvement on the diagnostic side.
So basically, everybody has an opinion. And my opinion is that the specificity of phage problem is an overstatement. We're getting better and better at finding broad host range phages that cover genotype space for some target bacterium. And engineering is already taking some phage and altering it to be useful across different genotypes that it was incapable of infecting before through some tweaking. So I'm actually pretty optimistic that the specificity problem won't be a problem for much longer, even if the technology I'm studying but not a big believer in is cocktails. I mean, right, if you think about it, that's what cocktails are. You ideally are putting together some phages that cover genotype space because you've measured it across different genotypes of your bacteria, and then you might just be, that is your solution. You're going to deploy that. Let's see, yes?
Sorry, maybe in phage therapy. Is there any limitations or difficulty in phage therapy due to the human immune system? I mean, I could easily envision applying it to maybe problems in the guts or skin, but for example, for conditions like sepsis.
Yes. So great question. Glad somebody asked that. Is there any concern over human immunity recognizing phages that are deployed and targeting them with antibodies? And yes and no. So there is more attention to that now. You can measure it, but it starts to be in this realm of what we've seen in the pandemic is just because antibodies are produced doesn't mean they're neutralizing. Okay? And now provocative statement number 2 is that you evolved on this planet in an ocean of phages, essentially. They're on the dust particles in this room. You're listening to me talk. You're going to inhale some phages that might just be completely new to your body. They've never seen them before. If not in your drinking water or your salad that you ate at lunch, I mean, you're going to see phages. And I would expect, as an evolutionary biologist who knows very little immunology, is that why would your body waste its time attacking something that you see all the time, including the entire history of the evolution of your species, if not all mammals on the planet, but let's not get into that quite yet. So that is where I have further optimism, but a lot of intense scrutiny right now is what is the human immune biology effect when you use this technology, and just more and more lessons learned every day. Not big deterrents, I would say. Yes?
How far apart, or what is the gap between phages and viruses that attack humans?
In terms of relatedness?
Well, I mean, is there a real enormous gap, or could phages actually start attacking humans?
Okay, so the question is, can phages start attacking human cells? I would admit to trying this in the laboratory, except it would be called a gain-of-function experiment, and the police might be waiting for me outside the door. So let's just say for the face, I've never tried it. Nevertheless, our collaborators and I are seeing something that others are, is that certain phages will enter your cells pretty readily. But they won't necessarily, well, no, not that anybody has ever observed, they won't replicate. Why it is happening, is it just passive? Is it actually that your cells take them up, sort of break those nucleotides apart maybe and use them for purposes of a resource within the cell? Certainly the phages we've deployed in the clinic differ in their capacity to enter human cells as measured in the laboratory, which I think is pretty cool, and it is an angle that biotechnology could go. Maybe I want to use phages to solve some problem within your cells, and I don't care necessarily about them killing bacteria. So once this is observed, can I leverage it for sort of a new way of medical technology to go? But that's a digression. I've ignored some hands here. Yeah, you.
So it sounds like a lot of researchers are talking about kind of like having a Rolodex of phages and kind of picking one that we think is going to impose a trade-off condition. Like, is there a sense for leveraging phage evolution, whether like in vitro or in vivo, could develop that library more, like allow targeting a specific thing? And also, I guess some of the last question, like, are there any risks with phage evolution where we'll think that, you know, like human cells it could, or it could like maybe just evolve away, maybe it starts affecting like gut cells that are, like gut pathogens that are like not.
Yeah, I guess I'll go with that last remark first. So there could be some phages that emerge more easily on your gut flora if I'm deploying them in your gastrointestinal system. So imagine I'm deploying phages in any target organ, actually, or maybe even in the blood for a sepsis problem. And here's where I start to wonder, okay, that could be problematic, but why would it necessarily be the fact that I find some rando phage in a river and I use it for this, and it's going to outcompete the other phages that are already in your body, naturally occurring in your microbiome, that are interacting with those cells all the time? I think it is plausible. I would assert it's highly unlikely that I would just stumble into some mysterious wonder phage that could do this. And I only stand by that. I haven't seen that yet. I've not seen that in our work or anybody else's. So I think it is an interesting evolutionary problem to study, is like, what is the frequency with which a phage that's deployed can emerge to become established in the ecosystem of your microbiome? Anecdotally, never seen it in the clinic. When we deploy the phages, when we're done, we can't find them anymore. At least we're largely doing pulmonary infections, so lung infections, and also prosthetic joint infections, where this would be post-surgery. A problem occurs with your artificial knee or your aortic arch or something that's increasingly trivial surgery, that the bacteria infest that, and they're multi-drug resistant or pan-drug resistant. We go in with phages and we try and solve the problem. So I don't want to claim it could never occur. A different talk for a different day is I think it's more of a concern with what's called oncolytic virus therapy, where you're taking human viruses and you're using them to target and kill tumors. Where there, I think there would be profound selection pressure for those viruses that can already infect your cells to just emerge on your cells. So if anybody actually is here and studying oncolytic virus therapy, I'd love to talk with you about the ecology and evolution of that, which I think is profoundly understudied. Yes?
I was wondering which aspects of this problem, or are there aspects of this problem, you would say, where we were limited by the extent of quantitative understanding of some of the processes, as opposed to progress lies through generative representation of the trade-offs that evolution happens to operate on, but this is not yet time for.
Good. So problem number one is, can I even find a phage that does this? And so what I haven't admitted so far is some target bacteria that we need to deploy and use phage therapy because antibiotics are failing. We have a long, long library and a growing list of phages that are found in environments. We characterize them and we try to use them. And other target bacteria, we're finding like essentially the same phage over and over again. Not the same strain, but it's interacting with the same structure again and again and again, and we can't use the collection and characterization of phages to our benefit to find something new. Then we would really have to go immediately to engineering, okay? The other problem is, how do I know it's working? So I think this is what's plagued phage therapy historically, is that the role of the immune system and whether you're kind of boosting it. Because when I said that if you deploy phages in the inner human cells, they may not replicate, but they actually boost interferon production. So you start to get a systemic effect that is beneficial, but it had been understudied through the years, and now we're figuring that out. So what I'm getting at is pharmacokinetics. So we need better tools that if I deploy a phage in you, could I use something like, I don't know, MRI technology to have these labeled phages for me to understand where do they go? Are they going to the target, but also to off-target organs? What is going on with their movement around the body? If I deploy phages against pulmonary problems, you could find those phages sometimes in fecal samples. So I love that technology. I'm not working on it, but increasingly I'm trying to collaborate with people who are engineers and developing better tools for us to understand when you deploy it, and even if it works, how is it working dynamically? And then it gets quickly, ideally, to familiar territory for me. It's like what's going on genetically and genomically. Because that's what a little bit more of my expertise area. Yes?
Taking this away from humans altogether.
Okay, now you're talking. Dispense with humans.
Well, thinking about these things called harmful alpha blooms, where you have massive blooms, they're releasing toxins, which actually do affect humans and
Yep.
and they also, when they go to these blooms, huge number of
Yeah, so wow, a lot to unpack there. So basically the success we've had largely is through personalized medicine, which is not done at scale, and that's the whole problem with personalized medicine. You have to put it in a clinical trial, get volunteers to essentially align with your outcomes, then you can label the drug. So that actually hasn't happened yet in the USA and many other places. But could you solve environmental pollution problems with phages? Yes, in principle. The question is if I'm doing this to, I don't know, clean up a lake where somebody has purposely used a bioterrorist attack of releasing bacteria in that lake, and it's going to water supplies quickly in my city, could I use phages to my advantage to destroy them? Maybe yes, maybe not. So they will select for bacteria to be resistant to the phage, and maybe these trade-offs that I would care about in the human body don't matter anything in the environment. And these coevolutionary cycles that we don't see necessarily in humans start to, am I really running out of time? Wow.
Yeah. So maybe we should let Paul...
Sorry. I wonder, why is Boris standing up and approaching the stage?
We completely hijacked your presentation, but that's very much sort of KTP style talk.
Yeah,
But
no, I love it.
Maybe we'll let Paul
Stop.
finish. No, don't stop. But finish.
Yeah. Oh, I'm done. I mean, basically, here's the provocative thing that I didn't get to. How viruses have evolved on Earth is a little tricky. You know, you have these 3 domains of life, and was there an ancient virosphere at the time that Luca existed, the last unique common ancestor to all cellular life? And did you have these viruses essentially then tracking the domains of life from the beginning? Or is it today some cell in my liver is going to have an autonomous thing bud off from it, like a virus, and today establishes a new lineage of viruses of humans that attack liver cells? Okay, so the problem is those are not independent. They could be happening on the time. What I'll end with is that I have a profound, if not weird, fascination with the evolution of extinction. Because extinction goes hand in hand with speciation. And well over 99.9999999% of everything that has evolved as a species on Earth has gone extinct. So that occurs through what's called a background extinction rate that is pretty well known in cellular life of multicellular organisms. And the question is, in these major time periods of life that are often separated by what we see in the fossil record as mass extinctions, or if you lose 80% of the species in the ocean, or the age of reptiles ends, you know, at the KT boundary, and you've got suddenly all dinosaurs essentially going extinct eventually, except for birds. These are major changes in the biosphere for the prevalence of species. What has happened with viruses along the way? Are there rules by which viruses evolve that they would track this through time? And by that I mean some viruses have RNA genomes that mutate quickly. They tend to cause kind of infections that are brief but traumatic, whereas other viruses have very large genomes made of DNA, and they'll hide out in your cells and avoid being detected by your defense systems. Like almost all of you are infected by herpes virus. I hate to tell you that today, but there's your news. So I would love to have a time machine and access to metagenomics, even if I go back in time, and look at virus communities or the virusphere on the planet. And is there something about the rules of engagement between viruses and cellular life that even despite mass extinctions, things have been robustly present in the virusphere? You can't do that experiment. We're out of time, but if you have an interest in finding life off Earth, my opinion is look for viruses, because they're the most abundant thing on Earth, and it's a matter of, can we get a better detection system for finding a virus, even if we can't grow it on anything? And I would say yes. We have those tools, at least for carbon-based life, that we can recognize. The problem is, if it's not that, and you're looking for it in some other environment, you have to turn to other tools. I'll stop now because Boris is looking at me threateningly, and thanks for your attention. Appreciate it.